My name is Sai Prasanna. I am going to teach concept dark reactions or light independent reactions under the chapter photosynthesis in higher plants. As we already know, photosynthesis, the site of photosynthesis is chloroplast. Photosynthesis has two reactions. One is light reactions. Those are light dependent reactions which are directly dependent on light, and the other which are called as light independent reactions. Calling light independent reactions as dark reactions is a misnomer, as these are not directly dependent on light, but they are dependent on the products of light reaction. It doesn't mean dark reaction doesn't mean that they are taking place in dark. It just means that they are not directly dependent on light, they are dependent on the products of light reaction. So let us recall three important steps which are taking place in the light reaction. The very first is absorption of light in the form of photons, which is taking place in thylakoid lamellae. As thylakoid lamellae has two photosystems, photosystem 1 and photosystem 2, which consists of pigments, those harvest light. And the second most important step is photolysis of water, which takes place in the thylakoid lumen, and that releases oxygen as a byproduct. And third most important step is formation of assimilatory power. What is this assimilatory power? ATP and NADPH. This is the most important step of light reaction, as this ATP and NADPH are utilized for dark reactions to reduce this carbon dioxide to glucose, or to fix the carbon dioxide to glucose, or for carbon assimilation. Yes, now let us see how carbon dioxide is reduced to glucose, how this carbon molecule is being fixed. This takes place in three cycles. One is C3 cycle, C4 cycle, and CAM cycle. If you take C3 cycle is also called as Kelvin cycle, named after the scientist Melvin Kelvin, wherein he utilized C14, which is a radioactive isotope, on an algae called as chlorella and conducted algal photosynthesis, wherein he discovered that the first stable organic molecule which is formed is 3-carbon compound, that is 3-phosphoglyceric acid. That is why it is called as C3 cycle, as the first stable compound formed is 3-carbon compound. And this C3 cycle takes place in all the photosynthetic plants. The next cycle is C4 cycle, which is also called as Hatch-and-Slack pathway. This is also called as Hatch-and-Slack pathway, and this is called as C4 cycle because the first stable organic intermediate form is oxaloacetic acid. It is oxaloacetic acid, which is a 4-carbon organic acid. This is converted to malic acid or aspartic acid by the enzyme malic acid dehydrogenase. This occurs only in C4 plants. What are C4 plants? Those plants occur in most of the tropical climates or hot climates. Okay. And these are mostly monocots. For example, sorghum, sugarcane, maize. Okay. And the only dicot which conducts a C4 cycle is amaranthus. I hope the introduction of C4 cycle is clear to you. Now we will see CAM cycle. What is this CAM cycle? CAM cycle is called as crustacean acid metabolism, and here also the first stable organic molecule formed is oxaloacetic acid. This takes place in crustacean family members, and these plants are succulent plants. These plants are succulent plants. See here I wrote. These plants are succulent plants, and they are seen in the desert areas. For example, cactus, agave, opuntia, and aloe vera. Few of the tropical epiphytes like orchids also conduct a CAM cycle. So now, as the introductions of three cycles are clear to us, let us enter into the in-depth biosynthetic pathways one by one. Let us start with C3 cycle. In this C3 cycle, there are three important steps which are taking place. One is carboxylation, second is reduction, and third is regeneration. Carboxylation is the most crucial step of this cycle as it is fixing carbon dioxide. And the first or the primary acceptor of carbon dioxide in C3 cycle is RUBP, which is 5-carbon compound. What is that? Ribulose bisphosphate. And this reaction is catalyzed by the world's abundant enzyme, that is Rubisco, ribulose bisphosphate carboxylase oxygenase. This carbon dioxide along with RUBP when catalyzed with Rubisco forms the first stable organic product in the Kelvin cycle, that is 3-phosphoglyceric acid. 3-phosphoglyceric acid. This undergoes reduction by series of reactions and forms two molecules of glyceraldehyde 3-phosphate, As RUBP is 5 carbon, carbon dioxide is 1 carbon. This forms a 6 carbon compound, right? But the first molecule is 3 carbon compound. It forms 2 molecules of 3 carbon compound and it gets reduced to 2 molecules of glyceraldehyde 3 phosphate. These 6 carbons, 1 carbon will be escaped to form the glucose chain. 1 carbon from each Kelvin cycle is escaped to form glucose chain and the remaining 5 carbons are regenerated to form RUBP. Regeneration is the most crucial step to continue the Kelvin cycle uninterruptedly. These two statements are very, very important. These are asked in previous years as well. Which is the most crucial step of Kelvin cycle? So, carboxylation. Which is the crucial step? So that the Kelvin cycle is continuing uninterruptedly means you have to say regeneration. Yes. So when 1 carbon is escaping from each cycles, as glucose consists of 6 carbons, how many carbons need to be escaped from each cycle? 1. So 6 cycles have to be conducted to form 1 glucose molecule. Now let us see the energetics of C3 cycle. To fix 1 carbon dioxide, it is utilizing 3 ATP, 2 in the reduction phase and 1 in the regeneration phase and 2 NADPH in the reduction phase. To form 1 glucose molecule, 6 cycles have to be conducted and 6 carbon dioxides are being utilized and it utilizes 18 ATP and 12 NADPH. I hope this is clear to you. There is one major drawback with the C3 cycle because of the enzyme Rubisco. Because of the enzyme Rubisco. What is the abbreviation or the full definition of this Rubisco? Ribulose bisphosphate carboxylase oxygenase. It means that it has equal potential to bind with carbon dioxide and it can also bind with oxygen, right? So whenever carbon dioxide and oxygen concentrations are equal, this Rubisco has higher affinity for carbon dioxide. It binds with carbon dioxide. But at high light intensity conditions, the stomata gets closed. When the stomata gets closed, there is no intake of carbon dioxide. Then the intracellular oxygen levels will be shooting up, wherein Rubisco will be binding with oxygen like this. Rubisco will be binding with oxygen. When it is binding with oxygen, it forms 1 molecule of phosphoglyceric acid and 1 molecule of phosphoglycolate, which is 2 carbon compound. This phosphoglycolate will revolve around 3 organelles. Those are chloroplast, peroxisome, and mitochondria. Okay, it revolves around all these 3 organelles and it will synthesize no sugars, it will synthesize no assimilatory power, rather it utilizes ATP for carbon dioxide evolution. So this process is said to be a wasteful process. This takes place only in the C3 plants only because of this enzyme Rubisco, because of the nature of this enzyme Rubisco. So what is photorespiration? Photo means light. In the presence of light, it is releasing out carbon dioxide by taking oxygen. Fine. I hope this photorespiration is clear to you. Now we will exercise few questionnaire with respect to C3 cycle. How much ATP and NADPH are required to reduce 4 molecules of 3-phosphoglyceric acid? To reduce 2 molecules of phosphoglyceric acid, it requires 2 ATP and 2 NADPH. Okay, to reduce it into 2 molecules of glyceraldehyde 3-phosphate. To reduce 4 molecules of phosphoglyceric acid, how much of ATP and NADPH are required? Of course, it is 4 ATP and 4 NADPH. Fine. Yes. Now we'll see the second question. Amount of assimilatory power required to form 1 sucrose molecule. You know how many carbons does sucrose have? 12 carbons, right? And how many does glucose have? Glucose. 6 carbons. Yes. So, if 18 ATP and 12 NADPH are utilized to form 1 glucose, which has 6 carbons. For one sucrose, which has 12 carbons, how much assimilatory power is utilized? Of course, it is 18 into 2 and 12 into 2, right? That 24 NADH. This type of questions are frequently asked, so that I'm exercising here. Yes. Now let us enter into C4 cycle. C4 cycle, as I have already told you, this happens in most of the tropical plants, those which live in high hot conditions. Yes, so these plants have a very good adaptation called as bundle sheath cells. If you can see here clearly, vascular bundles, and covering these vascular bundles, you have large cells which are called as bundle sheath cells, large cells with no intercellular spaces, with no intercellular spaces. These can be up to many layers, one to many layers around the vascular bundles. So this bundle sheath cells contain agranule chloroplast and they don't have any grana, and they are rich in rubisco. They are rich in rubisco. Clear? And the other cells is mesophyll cell. Okay, this trans anatomy will help them not to conduct photorespiration. So in C4 cycle, two carboxylations will happen. Two carboxylations will happen. Two carboxylations, one in mesophyll cells and the other in bundle sheath cells. In mesophyll cells, we have granule chloroplast. In bundle sheath cells, we have agranule chloroplast. So how these carboxylations are taking place, let us see in detail. Okay. So first, when carbon dioxide is entering into the mesophyll cell, it is converted to bicarbonate or this hydrogen carbonate by an enzyme carbonic anhydrase. This carbonic anhydrase converts carbon dioxide to bicarbonate or hydrogen carbonate. This reacts with PEP, phosphoenol pyruvate, which is 3-carbon compound, by the enzyme, catalyzed by the enzyme pepcase, phosphoenol pyruvate carboxylase. So what is this PEP? Primary acceptor in C4 plants. Primary acceptor in C3 plants is, or in C3 cycle is RUBP. Primary acceptor in C4 cycle is PEP. Very much important. Okay. And this fixes 4-carbon compound called as oxaloacetic acid. I already told you OA converts to either malic acid or aspartic acid by the enzyme malate dehydrogenase by utilizing one NADPH, or oxidized NADPH is oxidized to NADP plus. This malic acid or aspartic acid is decarboxylated and enters into bundle sheath cells, and it gets decarboxylated to 3-carbon compound and 1 carbon dioxide by an enzyme malic acid. Here again this NADP plus is reduced to NADPH and carbon dioxide. Okay, this 3-carbon compound, which is pyruvate, this is synthesized back to the mesophyll cells by pyruvate phosphate dikinase and utilizing 2-ATP. Here 1 carbon dioxide is fixed second time, means second carboxylation, as bundle sheath cells are rich in RUBP. Kelvin cycle is being conducted here, where for 1 carbon dioxide it uses 3-ATP and 2-NADPH, and extra 2-ATP are used by the mesophyll cells to generate pyruvate back to PEP, phosphoenol pyruvate. Okay, now let us see the energetics of C4 cycle. For the fixation of 1 carbon dioxide, 3 ATP are utilized in Kelvin cycle and 2 in the C4 cycle. Overall 5 ATP are utilized, okay, and 2 NADPH. For formation of 1 glucose molecule, the same story. 6 cycles need to be taking place. 6 cycles have to be taking place by utilizing 6 carbon dioxides and 30 ATP and 12 NADPH are utilized in C4 cycle. And C4 cycle is escaping the process of photorespiration as rubisco is present inside this bundle sheath cells. Rubisco is present inside this bundle sheath cells. Bundle sheath cells walls are thick. They are thick and they are impervious for gaseous exchange, means no gaseous exchange takes place. How this C4 acid is entering? By plasmodesmatal connections. What are these plasmodesmatal connections? This connects the cytoplasm. Of two cells. As this bundle sheath cells is impervious for gaseous exchange, there is no possibility of oxygen entering into the bundle sheath cells, as only carbon dioxide is rich. This conducts C3 cycle, the bundle sheath cells. So this is how it is escaping the process of photorespiration. I hope this is clear to you. And now we will exercise few questionnaire. Okay. Firstly, how much extra ATP is required to fix 6 carbon dioxide molecules? I already told you to fix 1 carbon dioxide, the extra ATP utilized by C4 cycle is 2 ATP. Now to fix 6 carbon dioxide molecules, how much extra ATP have to be utilized? 6 to 2 means 12 extra ATP are being utilized to fix 6 carbon dioxide molecules. How much extra ATP is utilized for the formation of 1 sucrose molecule? So here 6 carbon dioxide fixing means for 1 glucose, it is 12 ATP, right? 12 extra ATP means for 1 sucrose, the extra ATP will be 24 ATP. Such type of questions from the energetics will be asked for sure. That is why I am exercising in the class. Okay. Now we will see CAM cycle, which is also very important. Okay. CAM cycle, which is called as crassulacean acid metabolism. This is a photosynthetic adaptation of succulent plants like cactus, opentia, agave, aloe vera. Okay, wherein it gets periodic water supply. Water supply will not be throughout their lifetime. It is a periodic water supply. The plants which are belonging to arid regions and tropical epiphytes like orchids. And here in this plants, you will have scotoactive stomata. What is the scotoactive stomata? Stomata that opens during the night time and closes during the daytime is called as scotoactive stomata. In C4 and C3 plants, what you have? In C3 and C4 plants, you have photoactive stomata. Photoactive stomata. Okay. Yes, fine. Now, in this also, 2 carboxylations will take place. But as we have studied in C4 cycle, there are 2 cells, right, which are conducting carboxylations. One is mesophyll cell and the other is bundle sheath cell. But here it is conducting only in the mesophyll cells. 2 carboxylations are happening in the mesophyll cells only. Cells. But the period is different. One at night time and the other is at daytime. If you see, I told you, right, scotoactive stomata means it is opening at night time. At night time, when the stomata is being opened, carbon dioxide is entering, and with the help of the enzyme carbonic anhydrase, it gets converted to a bicarbonate, that is HCO3 minus. Again here, the primary acceptor is PEP. By the enzyme PEP case, it gets converted to OA. The same process. OAA will turn to malic acid and there it is going to bundle sheath cells. Here it is come to vacuoles. Here it is storing into vacuoles. Inside vacuole, malic acid will be stored and at daytime, when carbon dioxide is being released, this malic acid is released outside and this gets decarboxylated. This gets decarboxylated to carbon dioxide and pyruvate. This carbon dioxide will conduct Kelvin cycle and pyruvate will be stored as starch and it releases PEP whenever it is again starting of the cycle. Okay, I hope this process is clear to you. Now we will see this last chart which is very important and this is given in your NCERT. Okay, the cell type in which the Kelvin cycle takes place is of course in mesophyll cells. In mesophyll cells. The cell type in which Kelvin cycle takes place in C4 is bundle sheath cells. Here also in CAM plants it is mesophyll cells. Okay. Cell type in which initial carboxylation reactions are taking place in C3, though it is mesophyll. Initial carboxylation. Here also it is mesophyll. Here also it is mesophyll in CAM plants. Okay. How many cell types does the leaf have? Leaf have that fixes carbon dioxide. Only one cell type. Here you have 2. Here you have 1. Which is the primary acceptor? Here it is 5 carbon, that is RUBP. Here it is PEP and here also it is PEP, phosphoenol pyruvate. The number of carbons in the primary carbon dioxide acceptor. 5 carbons, 3 carbons and here also it is 3 carbons in CAM plants. Which is the primary carbon dioxide fixation products? Here it is phosphoglyceric acid. In C4 it is oxaloacetic acid. Here also it is oxaloacetic acid. Number of carbons in the primary carbon dioxide fixation products. 3 carbon, 4 carbon and 4 carbon in CAM plants. Does the plant have rubisco? Yes, it has rubisco. Yes, it has rubisco. Yes, it has rubisco. Does the plant have peptase? No, C3 plants doesn't have peptase. Yes, they have. And C4 and CAM plants does have peptase. Which cells in the plants have rubisco? Here it is mesophyll cells. Here it is bundle sheath cells. And here also it is mesophyll cells. Carbon dioxide fixation rate under high light conditions. Under high light conditions, so stomata is closed and there is no carbon dioxide. So it is low. Carbon dioxide fixation is low in C3 plants as photorespiration is taking place. Okay. Whereas here it will be high. Here also it will be high. Okay. And this CAM cycle is least bothered about light conditions because fixation happens during dark conditions, right? Fixation happens during dark conditions. Okay. And whether photorespiration is present at low light intensities? No. Okay. Photorespiration takes place in C3 plants at high light intensities, not at low light intensities. Here photorespiration is not happening. Photorespiration is not happening. Whether photorespiration is happening at high light intensities? Yes. Here no. And here also no. As they have an adaptation. Whether photorespiration would be present at low carbon dioxide concentration. Yes, whenever carbon dioxide concentration is low and oxygen is high, then only photorespiration is taking place. Here it is no, here also a big no. Whether photorespiration would be present in higher carbon dioxide concentrations. Yes, and here no, because they have an adaptation of peptides. Okay. Temperature optimum will be 20 to 30 degrees Celsius in C3 plants, 30 to 40 degrees Celsius in C4 plants, and here of course it is above 40, above 40. Okay, and examples for the C3 plant stats are legumes, tomato, okay, and those all are temperate plants, And C4 plants examples we already know, maize, sugarcane, and sorghum. CAM plants, we already know aloe, agave, orchids, cactus. Okay, so this biosynthetic pathways I have explained utilizing the NCERT diagrams to ease your understanding, to make your understanding easy when you are referring to the NCERT. Thank you. Let's meet in the next class.